as the sycamore tree blooms, it waits phoenix to come and perch.
On this note of famous Chinese proverb, welcome to the world of Sanyou's innovative drug discovery and technology platform, Sanyou's Super Trillion Antibody Library, STAL. This introduction footage will give you an insightful view of Sanyou's Super Trillion Antibody Library based on three aspects: phage display background, STAL's application and competitive advantages, and case studies.
So, from number one aspect, phage display background.
Phage display is one of the three mainstream antibody discovery technologies. This display technology won the Nobel Prize in Chemistry in 2018. As of the first semester of 2022, fully human antibodies accounted for 45.6% of the marketed antibody drugs. Among these, 25.8% of the marketed therapies were derived from antibody discovery using phage display technology.
Adalimumab is the first marketed fully human recombinant antibody drug using phage display technology. Since its launch at the end of 2002, the cumulated revenue up to 2022 has reached about $213.8 billion. The same therapy has generated sales revenue of about $21.24 billion for AbbVie in 2022. It was considered as a blockbuster biologic drug for 10 consecutive years. In addition to adalimumab, other marketed antibody drugs discovered through phage display technology include belimumab, the first antibody drug for treatment of SLE, and guselkumab, a new antibody therapy for psoriasis.
So, the number two aspect, STAL's application and competitive advantages. With the infrastructure of phage display technology, Sanyou Bio has developed a number of industry-leading super trillion phage display antibody libraries. By integrating with mammalian cell protein expression technology, Sanyou has formed a seamlessly integrated super trillion innovative biological drug discovery platform. Sanyou's platform integration is also equipped with fully automated screening technology. Our 9 super trillion antibody library can cover the following modalities: fully human antibodies, single-domain antibodies, mouse monoclonal antibodies, bispecific antibodies, polyclonal antibodies, targeted peptides, and targeted small proteins. The antibody discovery solution provided by Sanyou Bio is widely used in drug discovery, diagnosis, detection, and antibody customization for scientific research purposes. Our technology can assist our clients to discover candidates against rare and challenging targets, including drug discovery for development of challenging drug delivery methods. Modalities include ADC, RDC, CAR-T, BiTE, bispecific antibodies, and other innovative therapies. Sanyou Bio has a track record of more than 650 innovative antibody drug discovery projects for those of membrane protein, immune factors, multiple transmembrane proteins, and GPCRs. The successful rate for the discovery of lead antibodies is close to 100%.
Sanyou's STAL platform is in continuous iteration. Upon validation of the platform through numerous challenging projects, STAL got 3 major competitive advantages. First, STAL's core competency. We have large capacity, seamless integration to the validation with eukaryotic samples, and the drug developability guarantee. Now, industry's antibody libraries available in the market are of the capacity from tens of to hundreds of billions. Sanyou Bio's library power is of more than 8.5 times 10 to the 12th. Besides, our screened lead antibodies are second to none. Whilst with the antibody discovery platform and libraries currently available in the market, you can get as many as dozens to hundreds of lead antibodies. With Sanyou Bio Trillion Library, you can obtain from hundreds to thousands lead antibodies. The dynamic dual of library size and lead antibody generation greatly increase the chance of obtaining excellent antibodies. Moreover, the design of the library is based on the amino acid distribution of naive antibodies that are derived from a vast natural antibody repertoires. Whether single-domain antibody or traditional antibody modality, the expression level of more than 80% lead antibodies screened by Sanyou Bio platform can meet the requirement of drug development. Affinity kinetics of 608 antibody candidates obtained from Sanyou Bio's screening platform
were validated and analyzed. 61% of antibodies demonstrated affinity at the normal level. They can meet the high affinity requirements. The SEC purity of 1,982 candidate antibodies obtained from the screen platform were validated and analyzed. More than 73% had purity of over 95%, and 81% had purity of over 90%.
These data provide proof Sanyou Bio's super truly innovative biological discovery platform. Sanyou Bio's STAL is capable of discovering candidates for rare, challenging, and difficult druggable targets. STAL's second core competency, fulfillment of screening of all drug modalities with a short delivery time. The STAL platform libraries covers diversified repertoire, including immune, naive, recombinant, synthetic, and semi-synthetics. These libraries allow you to screen all sorts of drug modalities, such as single-domain antibodies, monoclonal antibodies, polyclonal antibodies, target peptides, and target small proteins. Our high-throughput and one-step screening technology guarantees short screening cycle. Sanyou Bio's Super Trailing Library screening is convenient. It shortens the cycle to 5 to 6 weeks, comparing to those using hybridoma technology with a lead time of 5 to 6 months. Sanyou's upcoming platform launch is going to improve the screening lead time and shorten the antibody screening cycle to 21 days. The third core competency of STAL, adaptability to research and development of challenging GPCR targets. G-protein-coupled receptors play key roles in the mechanism of many common diseases. They have been a hot topic for drug research and development. Its unstable structure, complex conformation, difficult protein preparation, and obstacles found in screening functional antibodies make development against GPCR targets very challenging. However, Sanyou Bio's STAL platform is adaptable to various antigen immunization forms. Its screening strategies can offer guarantee on discovering lead antibodies of challenging targets. So, when it comes to case studies, Sanyou Bio's STAL platform plays key part in antibody discovery process. It has succeeded in various project deliveries. As of March 2023, 5 projects have successfully obtained IND approval through Sanyou Bio's STAL. Currently, dozens of projects are in different research and development stages, and their molecular forms are various. Let's explore 2 cases. Case number 1, Claudin 18.2. Claudin 18 is a multiple transmembrane protein that constitutes tight junction structure with 4 transmembrane domains. It has 2 splice variants, Claudin 18.1 and Claudin 18.2. Their amino acid sequence homology is as high as 91%. Due to Claudin 18.2's highly selective and stable expression in malignant solid tumors, it's expected to become an innovative target for the first-in-class treatment of gastric cancer. The development of drugs targeting Claudin 18.2 is challenging because the antigenetic protein has complex structure of multiple transmembranes. Besides, they have to avoid antibody binding to Claudin 18.1. Sanyou Bio has developed single-domain antibody C2 from STAL. The C2 antibody showed comparable in vitro ADCC and CDC activities to the control antibody drug, as well as better in vivo anti-tumor drug efficacy. Case number 2. This project was for GPCR target. Through immunization with VLP and cell lines and construction of a mouse immune library,
Sanyou Bio has cross-screened and obtained lead antibodies, and more than 70% of these antibodies showed better in vitro binding activity than the control antibody. In addition to these projects, Sanyou Bio has a track record of experience in dozens of preclinical discovery projects for innovative antibody drugs, and hundreds of project delivery experiences in different drug research and development process. More than 600 companies have repeated business with Sanyou Bio. Some clients have established continuous business partnerships with more than 10 projects in development. Whether it is to build more types of libraries to screen more types of macromolecules or to overcome more challenging targets, Sanyou Bio aims to explore and provide more possibilities for drug research and development process to meet clinical needs. Turnkey solutions are required to make a breakthrough in current evolution of drug life cycle development.
A tall tree provides no shade, just as a tree does not form a forest. As this note of a famous Chinese proverb goes, Sanyou Bio aims to build partnerships with biopharmaceutical and biotechnology companies by providing our technology platform services to contribute value to the drug development process and life quality of the humanity.